Hi guys, we are Group 7 and our immunology lecture is on organ-specific autoimmunity. So starting off with what is autoimmunity? Autoimmunity refers to the presence of antibodies directed against normal components of a person known as autoantigens. These are also called self-antigens. An autoimmune disease occurs when autoimmunity causes clinical damage to cell tissue or disrupts the tissue's normal function. So you have an autoimmune disease when these anti-self antibodies start losing the function of an organ. Autoimmunity does not always result in an autoimmune disease. Every person is known to have low levels of anti-self antibodies responding to these autoantigens. It is only when a person has these high levels of anti-self antibodies, and again, they induce that clinical damage to an organ or a self tissue.
Okay, everyone, so talking about autoimmunity, we have to talk about the two types of autoimmunity that we have. We have systemic and organ-specific autoimmunity. Systemic autoimmunity refers to self-antigens that are present in autoimmune diseases that target and affect multiple organs, organ systems, and/or tissues. This is a more generalized disease that affects multiple organs within the body, such as in systemic lupus, which is depicted in the picture to your right. Organ-specific autoimmunity refers to self-antigens that are present in autoimmune diseases that target and affect one main organ or one organ system or one group of tissues. This is a more specified disease that affects usually just one organ, such as in the thyroid in Hashimoto's thyroiditis, which is depicted in the picture to your right.
So the outcomes of organ-specific autoimmunity have been divided into three different outcomes. The first one is a slow destructive process. This is when normal parenchyma is attacked by lymphocytes and antibodies and is replaced with connective tissue. So the image to the left just shows you different ways that the cell can repair itself due to cell damage. And I highlighted in the blue box where it says tissue repaired with diminished capacity. So this connective tissue is going to protect the organ from the damage, but it's also going to lead to a loss of function. The second outcome is a stimulating process, and this is when antibodies react with hormone receptors on the cell surface, mimicking the hormone's effects. So this is when the antibody acts as an agonist on the cell receptor, and it can cause an excessive production of peptides, chemical mediators, and growth of the target organ. The image to the left is Graves' disease, an autoimmune disease where autoantibodies, binds to the receptor, the TSH receptor, and stimulates the hormone synthesis. So in the end, you have an unregulated overproduction of thyroid hormones. The third outcome of organ-specific autoimmunity is a blocking process. So this is when a non-stimulatory antibody competes with other hormones that bind to these receptors. So this causes a gradual atrophy of the target organ, and the picture to the left kind of depicts this. So in this autoimmune disease, you have antibodies that are binding to the muscarinic acetylcholine receptors, and it blocks the function of the neurotransmitter. So this results in the paralysis of the muscle cell.
When talking about autoimmunity, we have to talk about this thin line of tolerance. We have two types of tolerance, central and peripheral. Central tolerance is the ability of the immune system to recognize self versus non-self. This is where we talk about negative selection of T and B cells in primary lymphoid tissues such as the bone marrow and the thymus. This is also where we talk about the AIR transcription factor, but we will discuss that in further detail later in the presentation. Peripheral tolerance is the ability of the immune system to continue and maintain this distinction of self versus non-self and prevent overreactivity of the immune system and its responses. This happens out in the periphery and usually is in the secondary lymphoid tissues, such as in the lymph nodes and spleens, where B and T cells have not fully matured from naive.
In relation to the mechanisms of organ-specific autoimmunity, we narrowed it down to three cells that play an important role in this organ-specific autoimmunity. So the picture on the left, it starts off with the antigen-presenting cell presenting the autoantigen to a naive T cell, which further differentiates into all these different kinds of T cells. But we're going to be focusing on Th1 cells, Th17 cells, and cytotoxic T cells. So the first cell we're going to talk about are Th1 cells, and starting off with Th1 differentiation, after autogen presentation, naive T cells are going to bind to interferon gamma and interleukin 12. So interferon gamma and interleukin 12 are being secreted by dendritic cells, macrophages, and natural killer cells. So these cells have determined that there is a pathogen or there's an antigen present, and because it's an autoimmune disease, it's an autoantigen that they were not able to distinguish self from non-self. So these chemokines are going to bind to this naive T cell, and they're going to stimulate the differentiation by an amplification of these chemokines. So these two chemokines are going to bind to the receptors of a naive T cell, and interleukin 12 is going to produce STAT4. Interferon gamma is going to produce STAT1 that is ultimately going to produce T-bet. These two are transcription factors that are going to amplify the production of interferon gamma even further. So interferon gamma is undergoing a positive feedback loop where it creates more to differentiate more naive T cells into more Th1 cells. So in the end, it's the positive feedback loop of interferon gamma that's going to create this upregulation in Th1 cells to fight off those target cells that are presenting that autoantigen. So Th1 cells are generally involved in the clearance of intracellular pathogens. They commonly produce interferon gamma, tumor necrosis factor alpha, and interleukin 2. They stimulate cytotoxic T cells as well, and B cell antibody production and macrophage activation. They are involved in cell-mediated and delayed type hypersensitivity. So the picture to the right just kind of shows you, after the presentation of antigen from the dendritic cell, how these Th1 cells kind of have all these different functions. So ultimately, an increase in Th1 cells due to autoantigen will induce interferon gamma, interleukin 2, and TNH alpha. Something we didn't really talk about was TNH alpha. This is going to induce inflammation and promotes extravasation of white blood cells into tissue spaces. And these images on the right just kind of put it all into pictures and sum it up so you can see where the different chemokines kind of correlate to the upregulation of specific cells. And the picture to the far right shows you what TNF-alpha really does and how it can promote the extravasation of those white blood cells into the tissue spaces.
Just as Yolanda talked about Th1 cells and cytotoxic T cells and their role in autoimmunity, we have to talk about Th17 cells and their role in organ-specific autoimmunity. Th17 cell differentiation occurs after an autoantigen presentation to a naive T helper cell that binds to TGF-beta and interleukin-6. TGF-beta and interleukin-6 produce transcription factors ROR gamma T, ROR alpha, STAT3, and IRF4, which aid in the differentiation of Th17 cells into fully mature Th17 cells that are ready to secrete pro-inflammatory cytokines. Th17 cells play a role in host defense against extracellular pathogens by mediating the recruitment of neutrophils and macrophages to infect tissues. They also play a role in tissue inflammation, immunopathology, and autoimmunity. They may be pathogenic in autoimmune diseases because of the pro-inflammatory cytokines that they release. They result in inflammation, cancer, and autoimmune disease due to tissue damage. The pro-inflammatory cytokines that Th17 cells release include interleukin 17A, interleukin 17F, and interleukin 22. These pro-inflammatory cytokines, when bound to certain cell receptors, activate a signaling cascade within the cell, which induces the production of chemokines. We learned about chemokines in earlier lectures of the semester and how they act as chemoattractants, which call in autoimmune cells such as monocytes and neutrophils to help and cause an increase in tissue inflammation. And we all know that there is a thin line between good inflammation within tissues and bad inflammation when not regulated properly.
Another T cell that promotes organ-specific autoimmunity are cytotoxic T cells, and we've talked about these before earlier in the semester. So cytotoxic T cells, they recognize infected cells via MHC class 1, And in relation to organ-specific autoimmunity, the recognition of autoantigen by the CTLs, they trigger degranulation and Fas-to-Fas-L interactions. So breaking down these mechanisms, CTL degranulation, the mechanism in that is that perforin monomers, they form a channel for granzymes to enter the target cells. So they form this kind of tube-like structure on the target cell, and it allows the granzymes to enter the cell and activate the apoptotic caspase pathway. The second mechanism of CTLs is the Fas-to-Fas-L interaction that it also induces the apoptotic caspase pathway. So all nucleated cells express Fas receptors. This is also known as CD95, and the CTLs express the Fas ligand, which is also known as CD178. So once these two Fas-to-Fas-L interact, again, it activates the apoptotic caspase pathway, and it creates the apoptosis of the target cell, which is going to promote that organ-specific autoimmunity.
We also have to talk about this negative selection and this air transcription factor that is expressed in the medulla of the thymus. We also talked about the air transcription factor in earlier lectures. This transcription factor provides instructions for making a protein called the autoimmune regulator. The autoimmune regulator plays an important role in helping T cells to distinguish the body's own proteins from foreign pathogens. A malfunction or disruption in the transcribing of air causes T cells to not be able to recognize self from non-self, which leads to the destruction of the body's own tissues and organs, which is what we refer to as autoimmunity. The air gene plays an important role in establishing tolerance within the immune system. In the picture below, A represents the normal homeostasis of our cells and how air is transcribed and regulated by Treg cells and how we get this tolerance established. B represents when we have a disruption or a malfunction in the transcribing of air and we are not regulated by Treg cells, which we get autoimmune diseases. Type 1 diabetes is a great example of an autoimmune disease because it is characterized by its own selective destruction of an individual's own insulin-secreting beta cells within the pancreas. The main characteristic of type 1 diabetes is the recognition of islet autoantigens by autoreactive CD4 positive and CD8 positive T cells and autoantibodies. This autoreactivity is due to a mutation within the air gene that we have just discussed before.
Here are our references, and here are our key points for the lecture. Thank you guys for listening.